Hey everybody, in this lesson we're going to talk about Punnett squares. So what is a Punnett square? So Punnett square is, it's basically a probability of inheritance. Now what does that mean? It's basically that inheritance is by chance. And that whatever you inherit, whatever trait you inherit is by chance. It's a probability that you inherit it. And the Punnett square is able to show that. Now how? Basically, it's used to predict possible gene makeups of offsprings. So here's an example of what a Punnett square shows. So you have two beans. You have a green bean, you also have a green bean. And both of them are heterozygous because you have a capital G and a lowercase g. So what do? It's sort of like Battleship, like ordered pairs. What's going to happen is this parent right here and this parent right here are going to give their genes over. So this G right here is going to end up with this one. While this G and this G will give big G, little g here, which will still be green. This box will be big G and little g here. And this last box is only the ordered pair in a sense, two little g's, which will be giving you, in this case, yellow shell color. And this shows you the probability that these offspring can inherit. In a sense that if you look at the genotype, for example, you see that you have the genotype homozygous dominant, you have the genotype heterozygous, and you have the genotype homozygous recessive. Now, with regards to that though, what you also see is the probability. These are 25% chances. So the two capital G's, the probability of offspring inheriting two capital G's is 25%. Probability of inheriting capital G lowercase g heterozygous is 50%. And then two lowercase g's is 25%. For the phenotype though, what you're going to notice is that there's only green and yellow. And what's the probability of inheriting those? Well, for the phenotype green, you can only inherit 3 of them, which is 75% chance. That means there's a 75% chance the offsprings will be green. And there's a 25% chance that the offspring will be yellow. And that's what we're talking about when we discuss Punnett squares. So let's try it out with this first one. In this first one over here, we have example 1. Black fur is capital B is dominant to white fur in mice. Cross a heterozygous male with a homozygous recessive female. And here are the alleles of both of them. So what's going to happen is I'm going to create a Punnett square. So you want to draw a Punnett square that looks like this. Now, so here's my Punnett square. Now, what we're going to see here is we're going to have to now put the parents' their alleles up. Okay, so we got to let's put the male parent right here and the female parent right here. And now we're going to write in each box their alleles that will show up for the offspring. So the first one we're going to have capital B, lowercase b, because that's what we have, capital B, lowercase b. Second one we're going to have is capital B, lowercase b. Third one up here is going to be 2 lowercase b's, and this one's also going to be 2 lowercase b's. So that's our Punnett square there. Now, if you take a look at this, what is the ratio of the genotype and phenotype? Well, let's take a look at the genotype first. What is the 2 different genotypes we have? We have a heterozygous, and we also have a homozygous recessive. What's the probability of each? Well, the probability of each is going to be 50% each. So 50% each. Now, when we look at phenotype, though, what will we get? Well, looking at this phenotype, what will capital B, lowercase b give you? Well, because capital B is dominant, that means this heterozygous will show dominant. So black fur will be 50%. Then what's the other one? 2 lowercase b's. That will be white fur. And that will be 50%. So that is my 2 ratios. my genotype and phenotype. Let's look at our next example. You have now two hybrid mice, and they're going to cross. Okay, so again, let's create our Punnett square. Okay, so there's our Punnett square. Let's create our boxes so we could do this. And there's our boxes. Okay, so we're going to have two parents that are both heterozygous. Heterozygous. So when we create and put these in, we have two capital B's here, a capital B and a lowercase b, a capital B and a lowercase b, and two lowercase b's. So if you look at this, what are my genotypes? Well, my genotypes here are going to be two capital B's, a heterozygous, and a homozygous recessive. What's the probability of each? Well, here, two capital B's only exist out 1 out of 4 of these boxes, so it's 25%. Heterozygous appears in 2 of the boxes, that's 50%. And then homozygous recessive is 25%. All right, now let's go phenotype. Okay, so we really only have two phenotypes, black fur and white fur. What are the phenotypes present? Well, let's look at black fur for a second. Black fur is going to be, as you can see, this one forms black fur, and this one also forms black fur. That's 75%. What's white fur? Only 25%. Only this allele can form white fur. All right, so these are my genotypes and phenotype. Let's look at this one now. A man and a woman having these alleles, both with brown eyes, have a child. What are the chances this baby will have blue eyes, which is 2 lowercase b's? Well, again, let's create our Punnett square. Right away, we know that because they're heterozygous, because both of these are heterozygous, so let me actually adjust this so it's a bit clearer. Because both of these are heterozygous, you know that brown eyes must be dominant in this case. So both parents are these alleles. So we're going to fill in our Punnett square. And which of these boxes will have blue eyes? The only one that has blue eyes is this one. And that one is only blue eyes, which is BB, is only 25% chance. That means the offsprings that are born from these two parents, there's only a 25% chance that they will have blue eyes. All right, let's look at how sex is determined in the baby, okay? So we talk about sex, we're talking about male or female based on their chromosomes, the XX chromosome and the XY chromosome. Because an average human, they have 23 pairs, which equals 46 chromosomes. And the 23rd pair is the sex chromosomes. Now, in the sex chromosomes, what we have is that they are XX is female and XY is male. The other chromosomes, the other 22 chromosomes or pairs, other 22 pairs of chromosomes are what we call homologous pairs. What homologous means is that they are the same size with the same genes. All right. Now, there's another actually a homologous one. The female chromosome is also homologous because you have two XX chromosomes. So what does that have to do? Well, if you look at this, these are what we call karyotypes. These 22 of them right here are what we call the homologous chromosomes. They're the same size and they have the same, the 22 right here, they have the same genes in the same location. It's only these ones down here that we call the sex chromosome. They're the ones that determine the sex of the offspring. So if you look at this, what is the probability of a couple having a boy or a girl? Who determines the sex of a child? Well, let's do a Punnett square. We're going to do a Punnett square, of the sex chromosomes only. All right? So if I create my Punnett square again, you're going to see that in one side, let's put the XX chromosome here, and the XY. And if I complete this Punnett square, I'm going to get XX, XX, XY, and XY. So look at this. What's the probability of, for the male chromosome? The male, which is the XY chromosome, is 50%. For the female, which is the XX chromosome, is 50%. So it's a 50-50 chance that this can happen. But who, in a sense, not determines, but who passes on the Y chromosome? Only the male can pass on the Y chromosome because there is no Y chromosome in the female. So that is what we talk about when we talk about the sex chromosomes. But there are different types of genes in which that they aren't just dominant and recessive. And one type is what we call incomplete dominance. Okay? Incomplete dominance is defined as when one allele is not dominant over another. AKA, there is no dominant allele in a sense. So what will happen is they will blend. So their alleles, their genes, their trait will blend. For example, if someone has, like, let's say red eyes plus blue eyes, if they have those two alleles in them, they will have purple eyes, for example. So let's look at an example such as example number 6. So look at example number 6 here. What we're going to see is, we're going to see that we have two pink flowers crossing. Now, the first thing we got to figure out is pink. What colors make up pink? And it's red and white. What that means is that a pink flower has both the red allele and the white allele in them. So if we create our Punnett square for these two flowers, what we're going to see is both flowers have the red and white allele. Now, a lot of times, to differentiate them, what you can do is you can use the same letters, or because they're both not fully dominant, you can use different letters to represent them. So you can use R for red and W for white. So that means both parents, being pink flowers, is going to have the R allele and the W allele. The R allele and the W allele. So what would happen to Punnett square? You're going to have an RR here, an RW here, an RW here, and a WW here. So look at this then. What is the genotype ratio going to be? In this case, you're going to have these genotypes, RR, RW, and WW. And because of that, you're going to have 25% chance that it's RR, 50% chance it's RW, and 25% chance it'll be WW. What's the phenotype going to be then? Well, the phenotype for these kind of questions is it's 1-to-1 correspondence, which means that RR is red, and that's 25%. RW is going to be pink, and pink is 50%. And then WW is white, and white is 25%. So that is the genotype-phenotype ratio of what we call incomplete dominance. If we talk about codominance, on the other hand, that's a different type of dominance now. When you hear codominance, you're hearing that they're both dominant. So multiple alleles are dominant. In this case, 2 of them. And because they're both dominant, they will both show or both express. So an example could be, for example, stripes. If something has stripes, it could be codominant. If something is speckled, like a chicken that has multiple different colored feathers, that is codominant. You could even say, for example, dots could be codominant because they both colors show, for example. If you have people have different eye colors, that could be codominance. So there's many different types of examples of this. Let's take a look at what happens if I have codominant examples. So you're going to cross a black feather chicken and a white feather chicken, and chickens can have codominance in the form of speckled, aka they have 2 different colors in them. If you don't know what I mean, Google speckled chickens. All right, so let's create our Punnett square. So if it's a black feather chicken, the alleles of black feather would be, let's put BB as those alleles. So you have BB for black. White feather chicken is WW. What are my Punnett square going to look like? It's going to be BW, 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 and BW. So now, if I look at my genotype phenotype, what is it going to be? Well, my genotype in this case is going to be BW only, and it's 100%. What's my phenotype? My phenotype here is going to be speckled, and that is going to be 100%. So that means there's no way, really, based on these Punnett squares, that the offsprings of these two chickens will have a white feathered chicken and a black feathered chicken. So let's take a look at our next type of example, which are what we call sex-linked traits. So, looking at our karyotype earlier, we know that we have X and Y chromosomes. But there's something that you might have not realized. And again, these are the sex chromosomes, which is that the X chromosome is much larger than the Y chromosome. So let's go back up to our karyotype. Do you notice right here? The X chromosome is gigantic, the Y chromosome is tiny. Remember, chromosomes are DNA. If a DNA is small, it won't have that many genes. It will almost have none. The X chromosome, being much larger, has a lot more genes, and because of that, it actually has a lot more traits involved. How does that affect our males? Because the males have the Y chromosome. What ends up happening is that due to males having only one X chromosome, okay, due to males only having one X chromosome, whatever gene is on it will show. So it doesn't matter if it's recessive or dominant. Doesn't matter if recessive or dominant. So it could be something bad, for example, and it will show no matter what. It could be something good, but it will show no matter what. Because for females to have two X chromosomes, now they have that dominant recessive. For females, two X chromosomes allow for dominant and recessive. So that means for a female, if they do have, let's say, a gene for, I don't know, super strength and a gene that for weak strength, right? In those X chromosome, and super strength is dominant, that dominant super strength will show, even though they have a weak strength chromosome in them. For a male, though, if that X chromosome has a weak strength chromosome, the Y chromosome cannot dominate over it. There's just no gene present there. And that's what we call sex-linked traits. So what are some examples, real-life examples of sex-linked traits? Well, there's a few real-life examples of this. One example, and the most common example, is colorblindness. Colorblindness is a sex-linked trait. And colorblindness doesn't mean that you see the world in gray scale. No, what colorblindness means is you cannot differentiate color. So what that means is you cannot tell colors apart. And the most common type is really, a lot of people have, is red-green colorblindness. AKA things that are red and things that are green to someone who's colorblind will look the same. There is other types, such as hemophilia, aka your blood will not clot. And that is a medical emergency now because your blood clots whenever you get a cut, so you don't bleed to death. So hemophilia is another example. So here's an example of colorblindness tests you'll see a lot of times. You see a lot of them, for example, look like that because they are that because you got red and green to differentiate them. There's also red and yellow, of course. And there are other examples, but these are colorblindness tests that you see a lot of times. So this is a 12, that's a 5, that's an 8, that's a 3, that's a 29, and that's a 15. So these are what we call colorblindness tests. Now for sex-linked traits, what do? Well, for these, because they're associated with the X chromosomes, for the alleles, the letters that we use X superscript to represent. So what that means is, for example, let's talk about colorblindness, right? If you talk about colorblindness and someone has normal vision, aka no colorblindness, they will be shown as X capital N. Capital N meaning dominant in this case would be normal vision. But that means X lowercase n would be colorblind. So that is an example of this scenario. All right. So a female that has normal vision but is a carrier of colorblindness marries a male with normal vision. Give the expected phenotypes of their children. So a female with normal vision but a carrier of colorblindness, what does that mean? Well, a female would have these alleles for their sex chromosomes. There will be XN and X lowercase n. Why? Because female, 2 X chromosomes, but one of the X chromosomes has normal vision, the other one has colorblindness. The male having normal vision, that will only be X capital N Y. Remember, males are XY chromosomes. So let's create a Punnett square for this scenario. What would be the phenotype-genotype ratio here? So in this case, the phenotype-genotype ratio here is going to be, so let's take a look. We have X capital N, X lowercase n, X capital N, and a Y. In our first box is 2 capital N, so X capital N, capital N. And our second box is going to be capital N, lowercase n. Third one is capital N, Y. And the last one is capital N, lowercase y. So if you look at our genotype in this case is going to be 25% chance for each of these. Because you're going to have an XN, X capital N, that's 25%. You're going to have an XN, X lowercase n, that's also 25% chance. You're going to have an X capital N, Y, that's also 25% chance. And you're going to have an X lowercase n, Y, and that's also 25% chance. But now look at your phenotype. In both these first two examples here, they're going to be female. But in each example here, both of them, they're going to have normal vision. So if you talk about normal vision female, that's 50%. Which means that there's no way the female offspring of these two couple will have colorblindness. But what if they're a male? In this case, this male would have normal vision. If this one would be colorblind. So in this case, normal vision male is 25% chance, but colorblind male is 25% chance. That means this offspring has a 1 out of 4 chance that an offspring born to them will be a male who's colorblind. That's what that means. So that means half the males born to this couple could be colorblind. So this is an example of that scenario. The last type of Punnett square we're going to deal with is what we call multiple alleles. What multiple alleles are where you have 3 or more alleles for a trait. And the most common example is blood type in humans. Because we have A blood type, B blood type, and O blood type. So that's an example of blood types we have. But here's the thing though. Even though there's multiple types of alleles, humans can only inherit 2 of them. All right. So how does multiple alleles work for blood? Well, first of all, we understand that there is codominant blood types, aka there are 2 blood types that are both dominant, and those are A and B blood types. If these 2 are codominant, that means O blood type must be recessive. So recessive must be O blood type. Now, these blood types are attached to a protein in a sense called immunoglobulin, and that is represented by I. So just like our sex-linked traits, we're going to write them like this. Where if you have A blood type, that's going to be represented as I superscript A. If you have B blood type, that's going to be written as I superscript B. And if you have O blood type, that's actually lowercase I. That's O blood type. Oops, it should be the other way around. So O blood type is lowercase I. And in this case, these two, because they're capital, is dominant over the O blood type. Now, what A and B blood type are the different antigens attached to that blood type. All right? But how does crossing alleles work then? How crossing alleles work is that really we have 6 different genotypes. Okay, let's talk about first of all A blood type. To have A blood type, remember you could inherit 2 of them. You must have these 2 types of alleles. IA, which makes sense. Having both of them being A's will be A blood type. Or if you have IA, lowercase I, because remember, this is dominant over the O blood type. With B blood type, you can have IA, IB, or IB lowercase I. Again, that's another type. If you have O blood type, that just means you have no A or B, which is 2 lowercase i's. But wait, that's only 5. I said 6 earlier. Where's the last one? The last one is AB blood type. Because A and B are both dominant, what if you inherit both of those alleles? AKA you inherit both IA and IB. That is AB blood type. So this is an example of A blood type, B blood type, O blood type, and AB blood type. And it's because of our codominance there. And this chart just shows you again the antigen, why they call AB blood type, and it just gives you a little information about blood donor types. Because if you have A or B blood type, you can donate to someone who has AB blood. If you're O blood type, you could donate to everybody, but you cannot, if you have AB or AB blood type, you cannot donate to an O blood type. O blood type is called a universal donor because you can donate your blood to anybody because those antigens are not present, but you cannot accept blood other than just O blood type. So let's look at the last one example of these kind of Punnett squares, which is what would the possible blood types of a child or children born to a female with type AB blood and a male with type O blood. Let's do our Punnett square. So if I do my Punnett square again, what I'm going to see is I'm going to see our female having AB blood type, which means that their alleles are going to be A, IA, and IB, and the male being lowercase I, lowercase I. If I fill in my Punnett square, this is what I get. IAIB, or IAI, IBI, IAI, and IBI. Now if you look at this, let's look at our genotype. We only really have 2. We have IA lowercase I, and we have IB lowercase I. And each one is 50%. Which means the phenotype, the offspring can only really have A blood type and B blood. And each one is also 50%. That means there's no possibility that the offspring could have O blood type, and there's no way the possibility that the offspring could have AB blood type. So that is actually a way you could actually do, for example, a paternity test for this, for example. So that's an example of something that is talk about blood type for offsprings. The last thing we're going to look at is pedigrees. Pedigrees you can think of as almost like a family tree that shows genetic traits. So they're basically graphic representations of traits in a family tree. So how do you read these? Well, To read these, what you are doing is you're looking at, for example, first of all, the circles are females, which means the squares are males. Oops. Horizontal lines equals, in a sense, mates, while vertical lines talk about offsprings. Okay? Shaded in equals has trait. So for example, the trait is colorblindness, they have the trait. And then not shaded is no trait. And what if they're partially shaded? Partially shaded is heterozygous in this case. So if you look at this right here, we have the parents right here, and they're connected by a horizontal line. Then we have the vertical line dropping down to the 3 offsprings. This female has the trait. This male right here is heterozygous, is connected to this female who doesn't have the trait. And then they drop off to this offspring, which has 2 males. One of them has a trait, one of them doesn't. All right? So that is an example of how pedigrees work. And here are some other ways that could show how these show what kind of things we have, like autosomal dominant. When this happened is that cannot be recessive as 2 affected parents. So both parents have the trait. In this case, if they have the trait, because one of them doesn't, if they both have the trait, the parents must be heterozygous for that to happen. And it shows you just examples of things like that. So for example, X-linked, how it works, and so forth. So pedigree is a good way to show how family trees and traits are passed down. As always, make sure you keep yourself safe and healthy, and I'll see you soon.